Hello, my name is Ashley Cooney, and I am from the University of Iowa. Today I'm going to share an exciting story with you called Increasing Saline Tonicity Enhances Gene Transfer to Airway Epithelia. Our lab focuses on gene therapy for cystic fibrosis. Although I'm not giving a background on cystic fibrosis, it's a genetic disease affecting multiple organ systems. However, the leading cause of morbidity and mortality in people with CF is chronic lung disease. Our goal is to develop a viral vector to carry the gene that causes cystic fibrosis, which is called the cystic fibrosis transmembrane conductance regulator, or CFTR. So again, a viral vector can carry the CFTR transgene or gene editing machinery. And our goal is to aerosolize this into the intrapulmonary airways of the lungs. In order to deliver a successful gene therapy reagent, we need to think critically about what the cell targets are. In the lung, the conducting airways are comprised of the trachea, bronchus, and primary airways. If you look at the image on the bottom of the screen, this is a cartoon depicting the surface airway epithelial cells. The cells that line the trachea and bronchus are comprised of secretory and ciliated cells. But importantly, the basal cells that line the basement membrane are the progenitor cells of the airway. Even below the basal cells lie submucosal glands, which I will touch on briefly later. We know that basal cells are the progenitor population of the airway. Therefore, to achieve long-term correction, we know that progenitor cell transduction may be necessary to cure CF lung disease with a single viral vector treatment. There are problems that come with translating vectors to therapy. Gene transfer, we know, is enhanced when you disrupt the tight junctions. However, the vehicle formulations that we've used in the past to disrupt tight junctions are an extra hurdle for FDA approval. Briefly, the tight junctions are the joining pieces between the secretory and ciliated cells at the surface airway. There's a concern that comes from the field when you open tight junctions. It could be dangerous in people who have CF. Chronic bacterial lung infections could pass through the airway cells and lead to a septic infection. But I wanted to ask the pressing question, what if opening tight junctions would remarkably increase progenitor transduction? Essentially, would the reward be worth the risk? So we asked, are there examples of agents that do disrupt tight junctions that are already used in people with CF? And we screened a few reagents, but we came across hypertonic saline as the answer to this question. Hypertonic saline is FDA approved. It can be nebulized, and for an aerosol treatment or an aerosol delivery, this is important. We know that it is safe for administering to people with CF. People with CF inhale hypertonic saline on a daily basis. It is cost effective. There is not much that's cheaper than salt water. And it's not detrimental to titers when mixed with viral vectors. So even if all of the other things were checked, if the titer was destroyed by mixing a formulation with a viral vector, you couldn't proceed. So I'm going to talk a little bit about the vehicles that we tested to disrupt transepithelial resistance. This is a way that we can measure tight junction integrity. Essentially, we look at we measure the ohms of the cells following application of different media. The media is listed on the bottom. USG, or ultra-CRG, that's the media that we use to culture primary airway cells from lung donors. And USG is maintained on the bottom of the transwell insert, and the top is dry. So it is an air-liquid interface that recapitulates that schematic that I showed on the first slide. So we compared USG to media such as DMEM. We looked at PBS++, which is plus calcium plus phosphate, and PBS-. We know that PBS-— does decrease the transepithelial resistance. And then we tested a few other agents, which I'm going to briefly describe, but I'm not going to go into detail on. LPC, or lysophosphatidylcholine, lysophosphatidylcholine is a tight junction is used as a tight junction disruption agent. It is a natural airway surfactant. EGTA is a calcium chelator. Doxorubicin is a proteasome inhibitor that we routinely use with AAV viral vector delivery. Surfactant is delivered to neonates, and therefore we were interested in evaluating it as a gene therapy reagent. But you'll notice the last bar is 3.6% sodium chloride. And I'm going to walk through when we mix an adenovirus expressing GFP at an MOI of 250 with each formulation, we applied this to the airway surface of our transwells for 2 hours. And 5 days later, we imaged these. With PBS we saw no transduction. We know with LPC and EGTA, which are known tight junction disruption agents, we saw some transduction. With doxorubicin and surfactant, we saw no enhanced transduction. And this is the first image where we made the observation that sodium chloride was enhanced with hypertonic, that adenovirus transduction was enhanced with sodium chloride. So we excitingly wanted to test this in a more rigorous manner. And so we used adenovirus. We measured adenovirus transduction and observed that it was enhanced with an increasing percent of sodium chloride in a dose-dependent fashion. Because adenovirus has a basolateral receptor, the CAR or CXADR receptor, we know that if you formulate adenovirus expressing GFP with DMEM on the surface of epithelia, we see little to no transduction. Simply mixing this with a 1% sodium chloride enhances transduction, and this is further enhanced with increasing percentages. I'll note here that people with cystic fibrosis who nebulize hypertonic saline use percentages of 3 to 7%, and therefore we focused our target range on these percentages. We quantified the GFP expression from these airway epithelial cultures, and that's the graph on the bottom. So we used flow cytometry, and we quantified the different percentage of sodium chloride mixed with AdGFP, and then we quantified on the y-axis the percent GFP from each culture. And we saw that we observed a significant difference at this stage. Importantly, these cells are a mixture of heterogeneous cell types, and we quantified the percent of basal cells, ciliated cells, and secretory cells. We saw when we mixed these with varying percents of sodium chloride, we achieved equal amounts of basal, ciliated, and secretory cell transduction in the cultures of the GFP percent positive cell type. We then asked, can you pre-treat, and will this be as effective as a co-administration? And we learned that timing matters. Transduction increases with longer exposure time. So I'm going to walk through the experiment that we did here, but basically we either pre-treated with sodium chloride or add GFP. So the final time point was 2 hours. So this is simply a 2-hour application. So on the top bar, if we keep 3.6% sodium chloride constant, and on the bottom, we will keep adenovirus GFP constant, and we will vary the time of the opposing treatment. So the top, if sodium chloride is constant, it's essentially having a sodium chloride pre-treatment. So the first thing we did was we put it on for 1 hour, and then we added GFP. We put it on for an hour and a half, and then we added GFP for a half an hour, 15 minutes, and then 5 minutes. We did the same thing with incubating with an adenovirus expressing GFP, and then added sodium chloride later on. The bars in green represent the time that AdGFP was present, and the bars in gray represent the amount of time that sodium chloride was present. We quantified this by flow cytometry. So on the x-axis, it will give you a percent GFP, and we can look at the sodium chloride constant versus the AdGFP constant. You can quickly see in both conditions, there's a time-dependent response, and the co-administration what gave the highest yield. So importantly, we need to show functional correction in a disease model. We next asked if we delivered the cystic fibrosis gene, CFTR, by an adenovirus, If we mixed this add CFTR with sodium chloride, will this also restore the anion transport defect in CF airway epithelia? I haven't gone into the background of what CFTR does in this disease model, but essentially it transports chloride and bicarbonate ions. So therefore, we can clamp them in something called an Using chamber and measure their short circuit current. And this is how we measure functional correction. We found that add CFTR restores the anion transport defect in the presence of hypertonic saline, and I'll walk you through that next. This is an example short circuit current tracing of CF cells in red and non-CF or wild type cells in royal blue. We can give interventions such as FNI or forskolin and IBMX, and this will activate the channel and cause it to open. And so you'll see a sharp rise in current as shown by the non-CF, and GlyH is an inhibitor of CFTR, so this current goes down. As you can see in the red, there's no response to either of these interventions. If you put add CFTR with no sodium chloride on the apical surface and the virus cannot get in, we see no change in the short circuit current. Excitingly, when we put the exact same amount of add CFTR on CF airway cultures simply with 3.6% sodium chloride, we now are seeing short circuit current levels that align with the non-CF levels. We repeated this over multiple donors, and indeed we saw that it was significantly enhanced simply by the presence of sodium chloride. So adenovirus is a robust viral vector. There are other vectors that are being evaluated for gene therapy as well. So we wanted to see if this application could work for other viral vector approaches. And indeed it can. Here I'm showing that sodium chloride enhances lentiviral transduction. We have a GP64 HIV, pseudotyped HIV expressing GFP. You'll normally see lentiviruses pseudotyped with VSVG. GP64 is just another envelope that has tropism for airway epithelia. If you put that on the cells alone, or if you add 4.5% sodium chloride, we see a striking difference. This was the same volume, the same amount of virus, and the only difference was some of the media was replaced with sodium chloride. And we saw a significantly enhanced amount of GFP in cultures that were transduced with lentiviral vectors. This tracing should look familiar, similar to what we just showed with the add CFTR. Now we're using a lentivirus that's carrying CFTR, again, pseudotyped with the GP64 envelope. And if you walk through each line individually over time, we can again measure the response to forskolin and IBMX, or the response to GlyH, which will open and close the CFTR channel. And the y-axis again is the short circuit current. The red line is a CF airway epithelial culture. The yellow line is a CF culture that was transduced for 2 hours with a lenti expressing CFTR. The blue line is non-CF, and the green line is a lentivirus treated with sodium chloride. Again, this was reproducible among several cultures, and we see that the sodium chloride treated vector on airway epithelia enhanced anion channel activity. AAV is another viral vector. It's seeking a lot of attention now for being a translatable vector. And these vectors are also enhanced by sodium chloride. We're testing one capsid type here, but we know that this works with multiple capsid types. And so perhaps this is going to have a profound approach for people doing AAV gene therapy by simply adding in a formulation with sodium chloride. We can see a drastic boost in expression that's pretty evident here by the picture. We know that AAV 2.5T has a strong background for knowing that it can transduce airway epithelial cultures, and simply a 2-hour application with sodium chloride in the exact same volume, the same amount of virus, now we see a boost in the AAV GFP expression that's quantified on the right graph by flow cytometry. I will briefly go over the functional results here. Similar to the adenovirus and the lentivirus, we see that there is a short circuit current response to AAV when it's formulated with sodium chloride that is significantly enhanced compared to unformulated vector, and this is comparable to non-CF levels of short circuit current. So this is a very exciting, simple approach to enhance gene therapy vectors across the board. But what's the mechanism? How is this working? We looked through a number of different approaches, and what we've come up with is that it seems to lean towards ionic strength. And ionic strength is the concentration of ions in a solution. And so we asked, what's the best way to test ionic strength as a mechanism for sodium chloride mediated entry? So what we found was we tested mannitol, which is an uncharged sugar, and NMDG, which is a charged sugar, or N-methyl-D-glucamine gluconate. And essentially, if ionic strength is the mechanism, we should only see enhanced transduction with NMDG, but not with mannitol. So we looked at the impact of ionic strength on viral transduction. So the 1 through 7% of sodium chloride that you see at the top are the same images from the first slide. But we also tested potassium chloride, and this also seemed to enhance in a dose-dependent fashion. But when we test mannitol, we saw no enhanced transduction as we increased the dose. But with NMDG, we did see an enhanced transduction. We quantified this by flow cytometry, as shown in the graph on the right. And in short, other ions could enhance. However, sodium chloride has a strong track record of being safe for using in people with CF. Next, we asked, in the viral vectors that are enhanced with sodium chloride mediated transduction, are they receptor dependent? I alluded to earlier that the Coxsackian adenovirus receptor, or CXADR, is required for ad entry, and this is specific for adenovirus type 5. There are various other adenovirus types. Ad5 is the most prevalent, and it's the one that's generally evaluated for gene therapy. There are other adenoviral vectors that are being investigated for CF gene therapy, or for gene therapy in total. And what I'm going to use here is AD21 as a comparator. AD21 does not use CXADR. In fact, it uses CD46, and we know that it's CAR, or CXADR, independent. Here's an image showing you if you formulate AD5 expressing GFP either alone or with sodium chloride on the apical surface, as I've described, we see enhanced transduction with sodium chloride. If we knock out the CXADR, the CAR receptor, from these airway epithelial cultures, and the image that popped up on the right is a small Western blot where we used CRISPR to knock this out. So we have a knockout, and then we have the scrambled here, and you can see we can detect CAR in the scrambled cells, but not in the knockout cells, just providing evidence that we did get protein knockdown. And excitingly, when we formulate AD5 GFP with 3.6% sodium chloride in the CAR knockout cells, we don't see any transduction. However, when we use AD21, which is CAR independent, it doesn't transduce well without sodium chloride, but it robustly transduces with sodium chloride. The graph that just popped up is flow cytometry quantification, showing that these numbers, the percent of GFP, is significantly enhanced with sodium chloride only in the presence of its receptor. Next, we tested AD5 formulation with sodium chloride, so we know it's receptor dependent, but the next step in entry is a low pH endosome. So does it still require a low pH endosome? We tested chloroquine and baflomycin A1, which both inhibit endosomal acidification. So the airway cultures, the transwells that we work with, were pre-treated with chloroquine or baflomycin A1 for an hour prior to transduction. And again, we saw that GFP is enhanced with sodium chloride, but when you treat with chloroquine and baflomycin, this was significantly reduced, suggesting that adenovirus still requires that low pH endosome step and enters through its original pathway. I'm going to walk through, again, this decreased transduction efficiency following chloroquine or baflomycin A1 inhibition suggests that a low pH endosome step is still required for ad entry. So I'm going to walk through the mechanism checklist here. I didn't show today for the sake of time, but we looked at a couple of other options. We asked, is this an osmotic effect? Does it just take up everything from the environment around it? The answer is no, we did not see that. We tested extracellular pH. This did not have an effect on entry. What we found instead was that it seemed to be an ionic strength, and it was receptor dependent and still required a low pH endosome. So all of this validation in vitro is exciting and important to do, but if this does not translate to an in vivo model, it's essentially useless. So the next question is, does this work in vivo? And I hope you can clearly see here that the answer is yes. So what we did was we intratracheally delivered an adenovirus that expresses luciferase, formulated either with PBS or 0.9% sodium chloride or 7% sodium chloride. We delivered this intratracheally and we imaged the mice by an in vivo imaging system, or IVIS, 5 days post-delivery. And this was in a 6 to 8 week old BALB/c mice. And you can see, we and others have reported that if you deliver adenovirus to a mouse, you'll see nasal expression. However, with sodium chloride, we now see that this expression moves to the lung, and we can see enhanced expression with the same amount of vector in more areas of the intrapulmonary airways. So our lab has a lot of experience in doing gene transfer in pig lungs. Pigs have a lung anatomy similar to humans, and it has been a reproducible model for us. And so the next thing that we did was to show gene transfer in a large animal model. So we asked the question, does sodium chloride enhance transduction in pig airways? So we intratracheally delivered adenovirus expressing GFP with either 0.9% sodium chloride or 5% sodium chloride. We changed to 5% here to pick a middle ground percentage to show efficacy at this range first. The lungs were harvested 5 days post-delivery. The schematic on the right is the standard operating protocol that we've developed to use to assess entire lung gene transfer. So you can see in the lung schematic, there is a proximal and distal trachea as noted by the PT and the DT, and the lung lobes are labeled as they are divided. So there are 6 lobes, and each lobe is further divided into 2 to 4 pieces depending upon size. And the lungs can be embedded and sectioned at that point, which allows us to give quantification of lung gene transfer. So after we harvested the lungs 5 days post-delivery, we looked for expression in the airways. We looked at the live tissue under a fluorescent dissecting microscope, and that's the images shown on the left. The PBS images were the best images that we could find of the GFP expression. The remainder of the lung was black. The 5% sodium chloride image, that is what the entire lung looked like. So as I showed on the previous slide, we divided the lungs up into various regions and we scored them independently. And so that is what is graphed on the right. So we scored 0 for no expression, 1 low, 2 medium, and 3 high expression. And you can see throughout the entire lung, there were high levels of expression. And this was significantly higher than the PBS treated pigs. What was most exciting to me is when we we embedded the tissues and sectioned them, and we took a cross section of what the GFP expression looked like. The top image is a 2x image, giving you an idea of what this transduction looks like across the board. And the 10x image below is from the 2x image, just at a higher magnification. And we show that we were successfully able to transduce conducting airways as indicated by the stars, but the arrow also indicates that alveolar cells were transduced. So we could potentially be hitting multiple compartments in this lung. We quantified the airway transduction, large and small airways. So I have not gone into detail about cystic fibrosis, genetic lung disease. However, we know that this disease starts in the small airways, and so they are also an important target for the lung. So using a sodium chloride formulation approach with a viral vector, we can reach both the large and the small airways of the entire lung. We did some immunostaining to show what cell types were transduced with AdGFP when formulated with hypertonic saline. The images on the top right show the AdGFP is from the vector. It is not immunostained. So the GFP was vibrant and it was expressed throughout the whole lung, as I've alluded to. So the red staining here is an alpha tubulin, and so that just shows ciliated cells at the conducting surface. So we showed, although I don't have arrows indicating here, you can look in the publication, there are both ciliated and non-ciliated cells transduced in the large conducting airways. But importantly, we saw high levels of transduction of basal cells, indicated by the arrows here. Again, the AdGFP is from the vector. The CK5, or cytokeratin 5, is a basal cell marker for the airways. And we showed in large and small airways that basal cells were readily transduced. I briefly mentioned submucosal glands at the beginning. I didn't go into detail about them, but I'd like to show you a couple of images here. We know that for cystic fibrosis, the abnormal mucus that comes from the submucosal glands, which is a major cause of the lung disease, the submucosal glands that produce this would be an important target in order to repair the mucus, the defective mucus production. You'll see here the AdGFP is in green, and the red again is CK5, because CK5 will also stain the basal cells of the submucosal gland. You can see here by the yellow in all three, these are three separate images all showing the same thing. They're all submucosal glands, and it was just so impressive to see so much submucosal gland transduction. And I'd like to draw your attention here to the center image of the center bottom image, the hollow arrow pointing, that is a submucosal gland duct. The mucus travels from the round portion of the glands up through the duct, and that's how it's released into the airways. Not only did we get high levels of submucosal gland transduction, we transduced ductal cells. This is a very important target for gene therapy for cystic fibrosis, and this is now achievable when formulating viral vectors simply with sodium chloride. I have a few takeaway points I'd like to leave you with. I've shown here that adenovirus, AAV, and HIV, or lentiviral transduction, is enhanced when you mix sodium chloride when you mix each vector with sodium chloride. The range is pretty specific in vitro. We learned 3.6 and 4.5% gave the most efficacious results. In the airway cultures, they are a sensitive model. We can go higher in vivo, and we showed that also. The images on the right, I hope that you take away that the virus alone or virus with sodium chloride is profound. We identified the sodium chloride mediated mechanism. It seems to be enhanced by ionic strength. The vector entry remains receptor dependent, and it still requires a low pH endosome for entry. We know that disrupting tight junctions with sodium chloride is unlikely to pose an additional risk of systemic bacterial infection of people with CF. This has a long safety track record. What this formulation could do is to enhance our current CF vectors through clinical trials, and it would have broad impact on many other genetic lung diseases. However, this could go far beyond the lung, and there are many other applications yet to be determined, and we are investigating those next. With that, I'd like to thank the group from the University of Iowa. We have a very large cystic fibrosis lung biology group, and we have very important members of our group who led to the publication of this work. I would like to thank my funding. I've had a lot of support from various organizations, and this work and the advancement of this work would not be possible without them. And with that, I would like to take any questions.